Hi. I'm going to start my talk by sharing with you a project that I'm working on. It's a graphic novel entitled Noli Timeri in partnership with Anita Doran and Steve Green. We started this project when I was on sabbatical in Paris, and the story starts off with a bacteria that is floating in the air from a distant continent, and it descends upon Paris. It gets closer and closer to our hero building, and it ends up landing on a tree. And this bacteria colonizes the tree, so it reproduces and spreads until the tree is fully infected. And then the bacteria begins floating from the tree into an apartment building, and it infects objects inside of this apartment building, and ultimately the apartment manager, whose name is Leroy. Once Leroy becomes infected with this bacteria, because he's the apartment manager, he ends up going into all of the other apartments to do work and leaves this bacteria behind, which ultimately infects everyone in the apartment complex. Now, this story is about the spread of a bacteria that, when it infects people, it doesn't make them sick. It's a beneficial microbe. And what it does, because this is science fiction, we can make this bacteria do anything that we want. And the symptoms that people get is they begin having a strong sense of déjà vu, and once they come into contact with other people that are infected, their memories begin to merge with one another. Now, you might ask, are all of the elements of this graphic novel fiction? And the component about the spread of a beneficial microbe is probably not. As everyone before me explained, the majority of microbes on Earth, the viruses, bacteria, and fungi, are beneficial to everything on Earth, and including microbes. The reason that we know this now is because the way that we study microorganisms has radically changed even since when I was a graduate student 20 years ago. When I was a student, I worked for a short time in a microbiology lab, and this is the way that we would study microbes. We would culture them on a petri dish. But today, if you wanted to understand the microbiology of any environment, you would look at the base pairs A, T, G, and C, and make comparisons among those base pairs using big data analytics to understand the ecology and evolution of those microbial systems that you've sampled. Using this new technology, we have learned not only that microbes that are on your body are important for your health and well-being, but we've learned a lot about environmental microbes, and particularly the microbes that are in the built environment, where all of us are going to spend 90% of our lives. We know that microbes that are indoors impact human health. And we're learning now, using this new technology, that the way that you design and operate a building, you can influence the kinds of microbial ecosystems that live indoors. And if you put these two known entities together, what it suggests is that we can operate current buildings and that we can design future ones to promote the growth of beneficial microbes and to inhibit harmful ones. In order to do this means that you really need to understand what drives the types of microbes that are in the built environment. So I've spent the last 7 years, I'm the director of the Biology and Built Environment Center at the University of Oregon, trying to understand what attributes of the built environment shape what microbes are inside. We know that microbes get into the built environment through mechanical ventilation systems, and that they also enter the built environment through open windows. Another force that brings microbes into the built environment are people. We all bring our microbes indoors, and our pets do as well. Once microbes get into the built environment, which ones survive and thrive are a function of all of the environmental conditions in the indoor environment. The types of materials, the temperature, the relative humidity, the way that we clean, all influences these microbial ecosystems. One of the first microbial ecosystems that we decided to study at the Biology and Built Environment Center is the hospital environment. And the reason for this is because hospital environments consume an extraordinary amount of energy. And they also require, in many countries, mechanical ventilation systems, and this is what's driving a significant amount of this energy use. And a second reason that we studied hospitals is because 1 in every 25 hospitalized patients gets an infection. A hospital is not a safe place to go, particularly if your immune system is compromised. So we worked with Providence Health and Services, which is a hospital in Portland, Oregon, to understand how ventilation influences the indoor microbiome. We looked at hospital rooms that were designed in this way, and in every room we put 2 air samplers, and we also measured airflow and temperature and relative humidity. This is a photograph of one of the hospital rooms that we studied. What you can see are 2 liquid impingers. That's how you collect microbes, one way to collect microbes in the air. And you can see that there is this ductwork that we put into place where we took the air that was coming into the hospital room and transferred it out down onto the ground level. And then we opened the windows in these operating rooms to see what would happen to the indoor microbiome. And this is what the data looked like from this. There's 3 different types of treatments. There's the mechanically ventilated rooms in blue. There are rooms where we open the windows, which are in red, and we also put samplers on the roof of the hospital, and those are in green. And the first thing that I want you to look at is the x-axis. This is a measure of the variety of microbes that are in the air samples. We were looking at bacteria. There are thousands of species in every sample, but this is a measure of evolutionary diversity. And what's important to note is that you have the biggest variety of bacteria in outdoor air, whereas the mechanically ventilated rooms look like a monoculture. You have a very low diversity of microbes. And then if you look on the x-axis here, what we did was we took every sequence that we sampled and we compared those sequences to a database of known pathogens. So we were not able to identify pathogens with great accuracy, but we said if the sequence is closely related to a known pathogen, then it's a potential pathogen. And what we found is that in the mechanically ventilated rooms, you had the highest proportion of potential pathogens relative to the window ventilated rooms and the outdoor air rooms. Now, if you took all of these data points and you put them into a statistical map where the data points that are closest together have more similar bacterial communities than data points that are farther apart, what we found, which completely surprised me the first time I saw this data, I had assumed that no matter how you ventilate a room, you're always going to have some microbes from the outdoor air, even if you mechanically ventilate them. But that's not what we found. We found that rooms that were mechanically ventilated had a completely different microbial signature than the outdoor air. And the reason for this is if you take those sequences and then again compare them to a known database, the outdoor air samples look like they have microbes that are what you find in dirt and on plant leaves, whereas the rooms that were mechanically ventilated have the types of microbes that are commonly found in the human mouth, in saliva, and on our skin. And that was the first time, this was published about 5 years ago, that I realized how important humans are in the indoor environment, that we're shedding our microbial cloud, and when we, depending on how we ventilate, we may or may not be removing that human microbiome cloud from the indoor environment. The next data that I'm going to show you is work that I've done with a client in Silicon Valley through my company Phylagen. And what this client is really interested in is understanding the relationship between the design of offices and how that impacts the health and productivity of office workers. So the first thing that they wanted to understand in their built environment is, does the way that we design our office environment influence the indoor microbiome? And if so, can we then have a new lens for understanding worker health and productivity? This was, we studied one building on their campus, which had 2 floors. And the first thing that we did was we looked at the different space types that they had in this building environment. So we looked at the kitchen environment, and what you're looking at is the relative abundance of different bacteria that are in the kitchen environment. And we compared that to the office environment and the restrooms. And what you can see is every type of environment in an office space has a very, very different set of bacterial species that are present in it. We found that in the kitchen, there were a lot of bacteria that are commonly associated with foods. We did this sampling in winter of this year, and it happened that during that time of year in this environment, mandarin oranges were being placed out on the countertops every day. In the office environment, we saw prevalence, and this is basically looking at people's desks. We saw a prevalence of bacteria that seemed to be very common in the indoor environment. And in the restrooms, we found the types of microbes that are very commonly found in the human gut. The next thing that they wanted to understand is, in their office environment, this client was trying to make a decision about whether to have, or there's discourse about whether or not to have an open office environment, which fosters collaboration, or a closed office environment. And what I had seen in my former work was that in a closed office environment, you see a strong spatial segregation in the microbiome of your neighbors. So you might have a very similar microbial signature to somebody whose office is right next to yours, but on the other side of the building, your neighbor would have a very different microbiome. And we saw something very different in this open office environment. The way that we studied this was, again, we looked at, for every person's desk, we looked at the surface of their desk, which again showed microbes that are common to what's in your mouth and on your skin. And then we looked at their chairs, which again have microbes that are common to the human gut. And in this open office environment, what we found is, if collectively you compare desks that are side by side, you see a very similar microbial signature. And if you compare desks that are on the opposite side of the building, you also saw a very common microbial signature. So together, this suggests that the spreading of microbes in the open office environment is quite prevalent. This client also was interested to understand how the way that they ventilated their building influenced the indoor microbiome. So what they did was first had us look at their business company, the office space, the way that they typically ventilate it, which they have 3 filters in their ventilation system. And then they had us take the filters out of their filtration system so that they could simulate natural ventilation in this environment. What we did, this is a network with 600 air samples that we took inside the office environment. We did this over the course of 2 weeks, and after running an algorithm that separates the different data points depending on the microbial composition, this is what you see. So again, the data points that are close together have similar bacterial communities, and data points that are far apart have very different bacterial communities. And this is with the different treatments that have been overlain. And so the main thing I want you to look at first when you look at this network is we sampled at 2 different time points. The top half of the network is during the week when people were occupying the office space. And the bottom part of the network is when there were no people in the office space. So this was another set of data that I've seen where you see a very strong impact of having humans present in the indoor environment and what types of bacterial communities are in the air. So again, looking at the top of the network, this is where you see all of these examples are when people were in the building, and the bottom of the network was on the weekend when you didn't have people in the building. Now, what we found that was really important from a design standpoint is that you see the strongest impact of the treatment, which is either using recirculated air or 100% outdoor air with no filters when the building is unoccupied. And what this suggests is that if an organization wanted to change their ventilation practices to flush out the human signature, a good time to do that would be when there are no people in the building. And this is very common at night. There's a practice called the night flush that can be used for this, or on the weekends. So again, from this data set, we found that the air quality from a microbiome context is going to depend on these 2 attributes: occupancy, so how many and when people are in the building, and ventilation. But this client also wanted to understand how these different ventilation choices were going to affect their energy use. And what we found here is, what you're looking at is energy consumption by the mechanical ventilation system on the x-axis, and the relative abundance of allergens that were airborne. And these are allergens that are both come from people that are shedding them in the built environment, and that are also coming from the outdoor air. And what we found is that regardless of whether or not the building was occupied with people, which is in the blue, or if it was unoccupied, which is in the pink, when you go from the recirculated air with filters to the 100% outdoor air with no filters, it's a win-win, where you dropped in the relative abundance of allergens, and you also dropped in the energy use of that building. So together, this data that I've showed you and what I've been thinking about for the last few years is this concept of, rather than asking, how can we keep microbes out of the indoor environment? That's what I was taught as a civil engineer in the 90s, was that we treated microbes like they were pollutants. Instead, what we should be asking, and we will be asking, is how can we operate current buildings and design future ones to promote beneficial microbes and to inhibit harmful microbes? And I think that this line of thinking is already not only capturing the imagination of architects and designers, but it's beginning to be implemented. And what we're seeing is that it's becoming universally accepted and recognized that we are the small minority of our self is human and the rest of it is microbial. We're beginning to recognize that the microbes in and on our body are fundamental to our health and well-being. And we're now beginning to think about how can we engineer and design our habitat, our human habitat, the built environment, so that we can promote health and well-being. Thank you.